All right. So let's, in the saga of the protein and enzymes, let's talk about. We looked at the previous slide where we looked at how the enzymes function in an optimal environment. Now the question is, knowing what we know about proteins, are enzymes also affected by the general environment? And your guess is correct. The enzyme activity can be enhanced or inhibited by changes in temperature and pH. Even chemical changes, meaning what kind of environment it is in. Is it hydrophobic or hydrophilic environment, or what is in the proximity, that can impact the enzyme function. Each enzyme, to the point we have identified, has optimal range, temperature range in which it can function. It also has an optimal pH range in which it functions. Case in point, let's look at the optimal temperature for human enzymes. So the humans, we know our body temperature is around 37 degrees Celsius. And you will notice that our enzymes have been formed fits the function. They have been designed to function at the most optimum at that temperature. If our temperature goes beyond that range, for example, after 40, you see a rapid decline in enzyme activity. So that's the stage when the body is kind of shutting down because of high temperature. But notice where the human being's enzymes will stop functioning, the thermophile bacteria, the bacteria that live in, say, oceanic vents, or in the hot steams, hot, or the geysers. In those cases, notice how the enzyme activity actually takes off and reaches its optimal at 80 degrees, 75 to 80 degrees. So this really shows how the enzymes have changed, and they're programmed, adapted to work in the best suited environment for them. Within the human body as well, enzymes have all variety of requirements and properties. For example, pepsin, the enzyme that is for the breaking the proteins in the stomach, it actually works at perfectly at the pH, 2 pH, which is very acidic in the stomach. And as the pH goes towards basic, its activity goes down. Whereas the trypsin enzyme, which is present in the in the intestine, where you have a lot of bile fluid, which is basic by nature, the enzyme actually activity starts picking up. And as you get to more and more basic, around 8, that's when the activity peaks. So notice that the enzyme is a perfect example of form fits the function. Form here means the physical three-dimensional form and its properties. It is uniquely designed to work in special places within the organism, or even within different organisms. Other factors that can impact enzymes are cofactors. Cofactors are could be metallic as well, and in many cases they are also organic. So if they're organic, they're called as coenzymes. So let's look at this. If a normal enzyme and there's a substrate present, it can it should normally attach and be able to function. However, these two are competitive and inhibitions. Let me just go back here. Okay. Yeah, so we are just talking about inhibition. So the cofactors, coenzymes, helped with the enhanced work of pH of the enzymes. Over here, we are seeing how we could have competitive inhibitors, which will mimic the substrate and therefore not let the substrate attach to the enzyme, blocking it. So this is called competitive inhibiting. And this could be like sometimes we talk about these some kind of toxins, say for example alcohol, or you could have certain toxins which can go and block enzymes. However, when you overwhelm them with water, or you could basically eliminate them by changing the conditions. The non-competitive inhibitors will bind to certain areas which are not the active site, and they change the enzyme in the process. These are really difficult to get rid of because now we are not even close. We don't have a way to inhibit them. So many times the poisons, neurotoxic poisons and things like that would be non-competitive inhibitors.